Well, let's look at question 3. The first part asks for a comparison, right? The command word is compare the effects of temperature on cellulase from the two fruits, right? So the command word wasn't described, right? But that was what quite a number of you did, right? You described the curve to, say, the banana cellulase first, then you described the curve for mango without really comparing the two, right? Showing without very clearly stating how they are similar or how they are different, right? So compare, so compare also requires you to have both similarities and differences, right? Which quite a number of you forgot about the similarities, right? So the answers are as such. I'm not going to read out the answers. You can read the answers yourself, right? Just a reminder that compare, right, not describe. So really, you should talk about the similarities and differences that you can see based on the shapes of the two curves, all right? And remember to QV. Then for part 2, let's break down the question a bit first. The question is with reference to the types of bonding in proteins, right? So remember, this is about types of bonding in proteins, right? How differences in tertiary structure of cellulase blah 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 could explain the differences in the activity shown in figure 3.1, right? So when you are talking about the differences in the tertiary structure, it is with reference to the types of bonding. So again, it's differences in the types of bonding in the tertiary structure, right? So you state those differences first, then you use those differences to explain the differences in the activities as shown in figure 3.1, right? How the banana cellulase has a higher optimum temperature, has a higher maximum rate of activity, right? Based on differences in the types of bonding in the tertiary structure. Okay, so the banana cellulase obviously is a bit more stable in higher temperature since it has a higher optimum temperature, meaning it denatures at a higher temperature, right, past 70 degrees Celsius compared to the 60 degrees Celsius for the cellulase from mango, right? So we are looking for how the types of bonding in the tertiary structure, right, give rise to this higher stability in high temperature. Right? So the most obvious or the best answer would be that the banana cellulase has got more disulfide bonds, which we know are not disrupted by high temperature, right, in the tertiary structure. Okay, of course, you can also mention how the other bonds, right, besides disulfide bonds, how the other bonds that there are more of them as well, right? So overall, there's just more extensive interactions between our groups, which makes the tertiary structure of the banana cellulase more heat stable, right, requiring a higher temperature with a higher kinetic energy to disrupt the more extensive bonding or to disrupt the stronger bonds in terms of the disulfide bonds, right? So that allows the active site of the banana cellulase to be less altered or to remain the correct complementary shape, right, at higher temperature to allow effective collision and formation of enzyme substrate complex at a higher rate since the temperature is higher, right, leading to the higher maximum rate of reaction as shown in figure 3.1. Right, so these will be the answers, right? So what quite a number of you did was you did not, right, really make reference to the types of bonding when you are talking about the differences in the tertiary structure, right? Some of you went on to suggest that the tertiary structure for the banana cellulase has got an active site that is of a higher affinity or more complementary to the substrate, which may not be wrong, but that's not what the question is asking for, right? The question is asking for differences in the tertiary structure with reference to the types of bonding, okay? And then, right, many of you did not have a complete answer because you did not mention the shape of the active site, how the active site is maintained or not disrupted in higher temperature for the banana cellulase, allowing for effective collision and formation of enzyme substrate complex. So that point on active site is missing in majority of your answers. Okay, then move on to part B. Part B has got these case studies about the 3 enzymes, right? Pectinase, silanase, right? There's also the cellulase, which was mentioned in part A, right? And then we are looking at 3 different features for comparison. You have the Vmax, Km, and the catalytic efficiency, which is Vmax over Km. Okay, so with this, right, part 1 asks for the explanation why ultrasound results in higher catalytic efficiency in the hydrolysis of pectin. So first of all, it's hydrolysis of pectin, right? So only pectinase is involved in part 1. Not your silanase, not your cellulase, but it's hydrolysis of pectin only, right? So as mentioned in the information on top, right, pectinase is the one that hydrolyzes pectin, right here, how you can see my cursor, right? So only looking at pectinase, why the ultrasound increase the catalytic efficiency, right? So the question already said that the catalytic efficiency is higher with ultrasound, and you're explaining why the ultrasound makes it higher, right? The question is not asking you to describe the effect on the catalytic efficiency. The effect is already mentioned in the question, right? So simply just quoting the values of 945 versus 759 and saying that catalytic efficiency is higher is not actually answering the question, right? So it is already higher as stated. The question is why, and the why, the reason must come from the information in table 3.1, because it's with reference to the information in table 3.1 explain blah blah blah, right? So your explanation must come from information that you see. So besides the increase in the catalytic efficiency, which the question already mentioned, what other things do you see, right, in the table? So you also see the Vmax, which is the same. You see that the Km is lower with ultrasound, right? So same Vmax, nothing much to say, right? But lower Km. This is where then you can use the lower Km to explain why catalytic efficiency is higher, right? So lower Km means higher affinity, as you have learned under your enzyme, right? So lower Km, higher affinity of the pectinase enzyme to its substrate, which is pectin, right? So that makes it easier for the pectin to bind, therefore higher rate of effective coalition and formation of enzyme substrate complex, which then makes the catalytic efficiency higher. Okay, so this is how you should be tackling the answer. For the majority of you, just look at the comments, the last point, right? Many of you simply mention, yes, the Vmax is the same, the Km is lower, and therefore Vmax over Km, which is catalytic efficiency, is higher. But this is not an explanation, right? This is just stating that, yeah, Vmax is the same and Km is lower. So therefore, Vmax over Km is higher. There's no explanation there, right? So yeah, so that's not credited. Yeah, that is simply kind of just stating the information given without explanation, without linking how the lower Km especially, right, leads to the higher catalytic efficiency. Okay, then for part 2, right, most of you actually know the answer, right? But QV, yeah, that's all I'm going to say, QV, all right? And lastly, for part C, the differences between allosteric inhibitors and comparative inhibitors. The question is specifically on allosteric inhibitor, right? So allosteric inhibitor binds specifically to the allosteric site, not simply a site other than the active site. That is the term you use for non-competitive inhibitor, right? But specifically allosteric inhibitor, it binds to the specifically allosteric site. So that's what I will be looking for. All right. The competitive inhibitors obviously binds to the active site, right? Because it has structural resemblance to the substrate, which the allosteric inhibitor doesn't have, obviously. Then the mode of inhibition, right? What the inhibitor does, right? Again, this one, because it's specifically allosteric inhibitor, so the mode of the action of the inhibitor inhibitor is also a bit more specific, right? It causes a conformation change to the enzyme as a whole, right? Not just the active site, right? So it causes a conformation change to the enzyme as a whole. And there is this specific term used for allosteric inhibitor, is that the enzyme now is changed to the inactive state, right? So that would be something that we're looking for, right? And when it is in the inactive state, the reason why there is inhibition is also because the active site is no longer complementary to the substrate, which prevents binding of the substrate. All right, so two things that I'll be looking for, yeah, conformation change of the enzyme overall to the inactive state, and also the active site no longer being complementary, right? For the comparative inhibitors, the comparison would then be, right, based on the action of the inhibitor. The comparison would be that it blocks the binding of the substrate to the active site by itself binding to the active site, or competing against the substrate, right, to bind to the active site. Okay, and just one thing to note, right, just want to explain something for allosteric inhibitor, right? If you look at this graph here, right, it shows you the action of the effect of the allosteric activator, right, as well as the allosteric inhibitor in green and red respectively. The curve in blue is the normal enzyme activity without any activator or inhibitor, right? Okay, so you can see, right, that with the allosteric inhibitor, right, it actually reaches Vmax, right? Unlike what many of you try to mention, because you are mixing up allosteric inhibitor with non-comparative inhibitor, right? Allosteric inhibitor has this or the allosteric enzyme, right, has this phenomenon known as cooperativity, right, in which when substrate binds to one active site, then it changes the conformation of the enzymes to the active state, and the other active sites also becomes more of a higher affinity, right, easier for the substrates to bind, right? So that's the cooperativity, right? So the allosteric enzyme has got two states, right, as mentioned just now, right? Inactive state, which is stabilized by the allosteric inhibitor, and also the active state, which is stabilized by both the allosteric activator as well as by the substrate itself. Okay, so now let's not consider activator, since the question is only on inhibitor. But with high substrate concentration, what happens, right? With high substrate concentration, then most, if not all, of the enzyme molecules will be bound by the substrate at any one time, which also means most, if not all, of the enzyme molecules are in the active state because of substrate binding and because of cooperativity. Yeah? So if most or all of the enzyme molecules are in the active state, then the allosteric inhibitor has no effect. That's why the Vmax can still be reached at higher substrate concentration. Okay, so this is a special thing about allosteric inhibitor or allosteric enzyme. Yes, the allosteric inhibitor is not a comparative inhibitor. The allosteric inhibitor does not bind to the active site. The allosteric inhibitor is not competing with the substrate. So this is not the substrate in high concentration out competing the allosteric inhibitor. That's for comparative inhibitor, right? But the allosteric enzyme has got this phenomenon of active state and inactive state. And the substrate binding causes the enzymes to be in the active state. So because of this phenomenon, right, the allosteric inhibitor can have no effect in high substrate concentration when all the enzyme molecules, or almost all the enzyme molecules, are bound with substrate and therefore are in the active state. Okay, so this is something that maybe you did not think too much about, because or you equating allosteric inhibitor with non-comparative inhibitor as if they are exactly the same. They are not the same, right? Not the same because the allosteric enzyme is a bit different, right? Unlike normal enzymes. Okay, so this part is something that you probably need to learn, right? Because many of you have written or have mentioned that Vmax will not be achieved with the allosteric inhibitor. That is true for non-competitive inhibitor, not true for allosteric inhibitor because of the nature of the allosteric enzyme itself. Yeah, okay. Right, so that is question 3. I hope you learned something, especially for the last part on allosteric inhibitor.